Good morning everyone. Today we are going to see about food biosensor. Last two classes we have seen about various heat exchangers used in the food industry and then various dryers used in the food industry. And last class especially we have seen about the extrusion technology, that is a special lecture because the extrusion technology also involves thermal processing. Then next lecture is about the recent measurement techniques in the food processing. Food processing industries used to measure particular toxic components, right. So, in that direction, first thing is, so I should be able to measure, right. So, how much toxin is there. So, based on that only any further processing can be done. So, to measure the particular concentration of the food spoilage enzymes and various proteins and organophosphate components, toxins. So, these food biosensors are used. So, in this lecture we are going to see about the working principle and what is biosensors and some of the applications in the food processing industry. Otherwise, the techniques used are based on for example, potentiometric mechanism, calorimetric mechanism. So, that we are not going to discuss about. So, here we are only going to talk about the biosensing technique, not for the mechanism in which it is built. Okay. So, if you are interested in knowing that in depth, so I request you to refer the proper electrochemistry book to get to know the exact mechanism. So, we are going to discuss only biosensors. So, the introduction is safety of food and environment is a concern of food technologists and health scientists. So, that is why we are here, because the course is about thermal processing, still we are discussing about the biosensors because it also leads to safety of food and environment. The strong need for rapid sensitive detection of different components of foods and beverages along with the foodborne and waterborne pathogens, toxins, pesticide residues with high specificity, right. So, the thermal processing is employed in the food just to handle with the foodborne as well as waterborne pathogens as well as toxins present in the food material, right. So, to do that, first of all I will be able to detect them, like how much quantity or how much concentration it is available in the food. So, the rapid and sensitive detection, this is very much important because already we have analytical techniques to measure them, right. So, what we wanted is, as the research is progressing, so we want everything in handy, right. So, handy as well as rapid. So, the rapid and sensitive detection of different components, foods and beverages as well as the foodborne, waterborne pathogens, toxins, pesticide residues with high specificity. So, that means, so we are very much specific about particular component. So, the sensor what I am using should not show me the other components which are present in the particular food. So, it needs high specificity towards the component measured. So, example is optical based sensing system that measure luminescence. So, luminescence in the sense when not heated, it emits light, right. So, based on which, based on some chemical reactions happen in the analyte, right. Due to chemical reactions or atomic motions or stress on the crystals, it emits light, but there is no need for to heat, right. So, that is nothing but a luminescence. And fluorescence, fluorescence you all might know. So, fluorescence is emit light which emits light under absorption of electromagnetic radiation. Electromagnetic radiation. radiation. So, we can call them as a for example UV light. When absorbing UV light, it emits the light. So, luminescence also I can tell as a category of fluorescence, but when not heated due to chemical reactions or the atomic motions or stress on crystals, it emits the light. And reflectance, you know, the light is reflected, and absorbance, you know, it gets absorbed, etc., are some areas of applications of optical immunosensor. Optical immunosensor. So, when immunological reactions happens, so it emits light, so that is measured, and after that it is getting converted into digital signal. So, in that way it senses how much analyte concentration is there in the food particle. So, this is one of the example for biosensors. Then immunological methods rely on specific bind of an antibody to an antigen. So, this is another example. So, as I said here, so we are telling optical based sensing system, right. So, which it senses the how much light is emitted, that is converted finally into electrical signal. So, when the light is emitted, we told that there is some chemical reactions, but in biosensor there may be some biochemical reactions. So, that is what is told here. Immunological methods rely on specific binding of an antibody. So, what is antibody? It is a protein in the blood or maybe other body fluids also, other body fluids of, maybe we can put it, vertebrates. Okay. So, these are all protein which is present in the blood, or we can call it as other body fluids of vertebrates. So, what it does? It does the recognition part of it. Recognition in the sense, it recognizes the unique part of the foreign target called antigen. So, it unique part of the foreign target. Okay. So, what is this unique part of the foreign target? That is nothing but your antigen. Okay. So, this is the protein, so which recognizes only unique part in the sense specific antibody recognizes only the specific antigen. So, that is what it is said, unique part of the foreign target. So, what is antigen? So, this may be any harmful substance, which enters the body, and maybe it also causes the body to produce antibodies. Okay. So, antibody is nothing but a protein, so which recognize unique part of the foreign target. The foreign target is nothing but a antigen. So, what is antigen? Antigen is any harmful substance which enters into the body. It signals the, or it causes the body to produce antibodies. So, this antigen gets binded into antibody. So, this immunological methods basically rely on this particular mechanism. So, this antibody may be monoclonal, polyclonal or engineered also. So, based on the antigen we can create a antibody, that is what the engineered antibody or engineered protein. Antibodies are basically a protein. So, detection of specific microorganisms and microbial toxins requires immobilization of specific antibodies onto a given transducer that can produce signal upon attachment of a typical microbe or microbial toxins. So, another detection method is the specific microorganisms and microbial toxins, right. So, for example, I wanted to detect microorganism X or the toxin produced by microorganism X, which requires the immobilization of specific antibodies. So, that means the proteins onto the given transducer. Transducer is a element which converts one particular signal into another signal. For example, whatever physicochemical signal is given to the transducer, it converts the physicochemical signal into electrical signals, then further it is converted into digital signals. So, what it is told? So, if I want to specifically identify the organism or quantitatively identify the microorganism X or the toxin produced by the microorganism X, which requires the immobilization of specific antibodies onto the given transducer that can produce signal upon attachment of typical microbe or microbial toxins. So, here microbe or microbial toxins are the antigens. Okay. So, this antigens where it gets bind, right. So, that is nothing but an antibody. So, that particular specific antibody is attached into the transducer. So, when the antigen goes and binds with the antibody, so definitely a physicochemical signal is produced given to the transducer. So, this transducer further converts the physicochemical signal into electrical signal. Inherent features of immunosensors such as specificity, as I told, particular antigen will bind into particular antibody. Sensitivity, sensitivity is nothing but how sensitive it is, like how quickly it can measure the physicochemical signal. And speed, ACE and on-site analysis. For example, I told there are conventional methods also available. There are conventional methods which are available. So, but conventional method are lab bound, right. We cannot carry with in hand, right. So, if I want to measure the glucose level while traveling in the train, then I will be able to do by using biosensor, but that not I cannot carry with me AAS. So, that is another important. So, ACE and on-site analysis can be made use for various applications. So, that is also advantage of biosensors. Biosensors present attractive, efficient alternative techniques by providing quick, reliable performance. So, that is where the biosensors are very much wanted in the food industry. So, we have also discussed in one of the lectures TTI, time temperature integrators. In that also we have discussed about the using amylase, how the microorganism detection can be done. So, that is also one kind of biosensor. So, food preservation maintains food at a desired level of properties, right, and or nature for their maximum benefits, right. So, it should not be spoiled. So, the properties should be maintained, should not be spoiled due to microorganism or enzyme reaction. So, that is desired level of, so maintaining the desired level of property is very much important. Each steps of handling, processing, storage, distribution affects the characteristics of the food, right. So, what we have mostly seen in previous lecture is handling and processing, but storage and distribution also there may be a chance for the contamination. So, which may be desirable or undesirable. Sometimes it may be a desirable change, sometimes it may be a undesirable change. Understanding the effects of each preservation method and handling procedure of foods is critical in food processing, which lead to the safe food, right. So, monitoring of safety and nutritional quality of food are very essential. So, then and there during processing, during handling, during storage, during distribution, every point of contact. So, we need to monitor the safety and nutritional quality. The conventional analytical techniques which are used to check this quality and safety are tedious, time consuming, require trained personnel. Quick, sensitive, reliable techniques for quick monitoring of food quality and safety is very much essential. That is why we are here to discuss biosensors. So, now what is biosensor? Their specificity, biosensor specificity from biological binding reaction. So, one I told us antigen, antibody. antibody, antigen. So, that is example, which is derived from a range of interactions that include antigen antibody. So, that by now you know what is antibody, what is antigen. Enzyme substrate cofactor. So, that means, so substrate in the presence of enzyme, it is converted into product. Cofactor is nothing but ions which increase the activity of the catalyst. Catalyst here is nothing but a enzyme, right. It is said that it is a biological binding reaction. It may be due to antigen antibody relation or enzyme substrate cofactor, that relation, or receptor ligand, right. So, the ligands comes and attaches into the receptor, same like antibody antigen. So, this has specific sites, right. The receptor has specific sites where ligand comes and binds with it, or chemical interactions and nucleic acid hybridization in combination with the range of transducer. So, in any biosensor, so you will be able to find one bio receptor, right. So, what is this bioreceptor? Bioreceptor, if you see this combination, antibody is the bioreceptor. If you see this combination, the enzyme is the receptor where substrate comes and binds and gets converted into product and product moves out. So, if you see ligand and receptor, so your receptor is a receptor where ligand comes and binds, right. Or chemical reactions and nucleic acid hybridization is also there. So, this is nothing but a bioreceptor component. So, which we call it as a recognition. Okay. So, then it is given to whatever physicochemical signal produced from the bioreceptor is sent to the transducer. Okay. So, which produces electrical signal. So, this is nothing but a recognition compound. So, then the next one is, this is nothing but a transduction. Okay. So, then I need to process this signal processing, electrical signal, I will be able to process the signal processing. So, then after that it is converted into digital signal, what we get to see in the digital display, digital signal. This is what overall happening. So, this bioreceptor, so in combination with the range of transducer. So, this transducer can take any type of signal. So, I told here physicochemical signal. So, it may be a optical signal, it may be a colorimetric signal, it may be a electrochemical signal, or it may be a piezoelectric signal, any signal, right. Range of transducers will be used in here in the transduction component. So, a biosensor can be defined as a quantitative or semi-quantitative analytical instrumental technique containing a sensing element of biological origin, that is nothing but a bioreceptor, which is either integrated within or is in intimate contact with the physicochemical transducer. So, this is separately attached. So, that is what integrated within or is in intimate contact. So, this bioreceptor can go inside the transducer as well. So, either integrated within or is in intimate contact with the physicochemical transducer. So, this is what whole mechanism of biosensor. I need to have one bioreceptor which recognizes the analyte, then it produces physicochemical, it is physicochemical. So, it produces the physicochemical signal. So, which is given to the transducer, which converts this physicochemical signal into the electrical signal. So, this electrical signal is further processed using signal processing technique, and which gives the digital signal, that is we get to see in the display. So, now we are going to see, because the transducer signal processing is not our hand. So, we are going to take care of this bioreceptor component. So, that means, so I need to identify which is the antigen, which is the antibody, which is the receptor, which is the ligand, which is the substrate or which is the enzyme. So, based if I could find out that combination, then easily I can put it into the bioreceptor module, which creates further either optical or piezoelectrical or colorimetric or potentiometric signal that will be given to the transducer and further process goes on. So, a chemical sensor transforms chemical information ranging from the concentration of the specific sample component to a total composition analysis into an analytically useful signal. So, the biosensor is derived from the chemical sensor. What chemical sensor does? Chemical sensor transforms the chemical information ranging from the concentration of the specific sample component to a total composition analysis into an analytically useful signal. So, this chemical sensor also usually contain one is chemical recognition. So, there it is a molecular recognition system which is called as a receptor and physicochemical transducer. So, based on the chemical sensor only the biosensors are derived. So, in the biosensor it usually contains the biochemical mechanism. Here it is a chemical recognition. So, here it is a biochemical mechanism interfacing the optoelectronic system. Optoelectronic system is nothing but a transducer and further processing. A device that uses the specific biochemical reactions mediated by a isolated enzymes, immunosystems, tissues, organelles or whole cells to detect the chemical compounds, usually be electrical, thermal or optical signals. A device that uses a specific biochemical reactions. So, those biochemical reactions were mediated by a isolated enzyme. This is what we have told in the enzyme substrate cofactor system. Immunosystem is nothing but antibody antigen. Sometimes it is not only this enzymes or antibody antigen system, it may be a whole microbial, we call it as a microbial biosensor, right. So, there we will get to see either tissues or whole cells to detect the chemical components, usually by electrical, thermal or optical signals. So, these were given to the transducer. So, what are all the prerequisites for biosensor, right? As I said earlier, selectivity. So, high selective for the target analyte. Target analyte is nothing but what we are going to measure and show minimum or no cross reactivity with the moieties having similar chemical structure, right. So, as I said, when antibody is a protein, so which antigen is a toxic component or maybe our target analyte which goes and binds in the antibody. So, if there are any similar chemical structure of moieties present in my food particle, so the antibody should not get confused to find out which is the target analyte or which is the moiety which has the similar chemical structure with the target analyte. So, that is very much important. And sensitivity. The biosensor device should be able to measure in the range of interest for a given target analyte with the minimum additional steps such as pre-cleaning and pre-concentration of the sample without doing any pre-processing. So, my biosensor should be able to measure in the range of interest for a given target analyte. For example, this particular concentration to this particular concentration without any additional steps. Then linearity of response. The linear response range of the system should cover the concentration range over which the target analyte is to be measured. So, that means if I am measuring with respect to electrode potential, so the potential versus concentration, right, should be in linear. So, that is what linear response range of the system should cover the concentration. For example, which concentration, minimum concentration to maximum concentration, it should give me linear relationship. Linear response range of the system should cover the concentration range over which the target analyte is to be measured. The next one is reproducibility of signal process. So, reproducibility already you know. So, how many ever times I measure, it should give me same response. So, that is nothing but a reproducibility. The next one is quick response time and recovery time, right. So, this is another reason why we go for biosensor. The biosensor device response should be quick enough so that the real time monitoring of the target analyte can be done efficiently. The recovery time should be small for reusability of the biosensor system, because how many uses it go, that is also depend upon the quick response time and recovery time. The recovery time in the sense, so after measuring one particular target analyte, how quick it comes back to the original position. So, that depends upon the recovery time should be small for reusability of the biosensor system. So, that means, so for example, one particular food I am measuring the low concentration of the target analyte, another food material I am going to measure the target analyte concentration in one sample and another sample is different, right. So, first sample if I am measuring, so it should recover to the original position so that it can be measured in the second sample, right. If the recovery time is high, then I need to wait, because or it may interfere the concentration of the target analyte in the second sample as well. So, the response time and recovery time should be very much small. The stability and operating life. As much most of the biological compounds are unstable in different biochemical and environmental conditions. Mostly biological compounds are very unstable. The biological element used should be interfaced such that the activity is retained for a long time, so as to make a device marketable and practically useful in the field. So, that is important. So, we told that bioreceptor, right, recognition. Before recognition, so your biological compounds should not go to other state, because we have seen that most of the biological compounds are unstable. So, that this unstable state should be high enough, right, to give the room for the biosensor to recognize or sense the biological compounds. So, that is also important, stability, right, or the device should be able to measure the particular biological compounds within their unstable state. So, that is also important. So, the key component of a biosensor is the transducer, which makes use of a physical change accompanying the reaction. So, what are all physical change? If heat output or absorbed, right, heat input or output by a reaction. So, that is nothing but a calorimetric biosensor. So, as I told, here I have a bioreceptor. So, here I have a transducer, right. So, based on which kind of signals I am giving to transducer. So, that is where here if heat output by the reaction, that is nothing but a calorimetric signal I will give. Signal will be of calorimetric. Okay. If there is a changes in the electrical or electronic output, that is nothing but a electrochemical signal I will be giving into transducer. If there is any redox reaction, so what we give is amperometric. So, that is called amperometric biosensors. And light output or light absorbance difference between the reactants and products are given, that is called optical biosensor. So, I may be giving optical signal as well. Based on mass of the reactants and products, we call it as a piezoelectric biosensor. So, in that case I will be giving piezoelectric signal to the transducer. The working principle is based on which signal or which physicochemical signal I am giving into transducer. It may be calorimetric, it may be electrochemical, it may be amperometric, it may be optical, or it may be piezoelectric. So, as I said earlier, we are not going into detail about each working principle, because it is just introduction of food biosensor, but we will see as much as possible. The electrical signal from the transducer is often weak with a heavy noise, right. So, to increase the signal to noise ratio, a reference baseline signal derived from a similar transducer without any biocatalytic membrane from the sample signal should be used. So, if you remember, here we have seen about the recognition, transduction and signal processing. So, we will see here So, what we told is bioreceptor component, right, or we can say this as a something like this. So, this is my reaction site or reaction pad we call it. So, here you have a substrate, right. So, here your enzyme is attached, this substrate converts into product. Okay. So, this is one particular example I have taken. So, what happens then after that, it creates any one of this reaction or this biochemical reaction is converted into any one of the signal. If you use optical biosensor, it is optical. So, if you are using electrochemical biosensor, it is a electrochemical signal, right. So, if you are using colorimetric, it is a colorimetric signal, or if you are using potentiometric, it is a potentiometric signal. Okay. So, all these signals are fed into transducer. Okay. So, after the transducer, it goes to amplifier, that is what they are saying. Okay. So, this is the amplifier. So, when it is given to amplifier, it is supposed to give me the, it is supposed to go to processor, right. From the processor, it will give the digital display, digital display. Okay. So, when it converts the optical or electrochemical or colorimetric or potentiometric signal, so this transducer produce the electrical signal. So, this electrical signal will be of weak and heavy noise, right. So, electrical signal will be weak with heavy noise. So, to avoid that, we are introducing a reference signal. Okay. To increase the signal to noise ratio, a reference baseline signal derived from a similar transducer without any biocatalytic membrane from the sample signal should be used. So, this is without any biocatalytic membrane. So, this reference signal is used in the amplifier. So, this is nothing but a amplifier. Okay. So, with this particular weak heavy noise signal is compared with the reference signal. So, that is the way the signal to noise ratio is increased in the amplifier. So, this is further go to processor. Processor is signal processing. So, it creates the electrical output, that electrical output is amplified, then given to the signal processing unit. From the signal processing unit, what we get is a digital display, right. So, now understood working principle is, so for example, I have a reaction pad, here I have taken the enzyme substrate cofactor biochemical reaction, the substrate, the enzyme is kept in the reaction pad. So, substrate comes and binds with the enzyme and converts into product. So, this particular biochemical reaction is produced as a electrochemical, optical or colorimetric or potentiometric signal. So, which is further fed into transducer. So, this transducer produces the electrical signal. So, that particular electrical signal is weak as well as heavy noisy. To avoid that or to increase the signal to noise ratio, so we are using a reference electrical signal. So, which is produced from the transducer without any biocatalytic membrane, right. So, then the produced electrical signal from the transducer is compared with the reference signal. This is processed in the amplifier and fed into the processor, which process the signal and gives the digital display. This is what working principle. The difference between the signal is very weak and amplified as a readable output in amplifier. The above process removes the unwanted noise from the signal. That is what happened in the amplifier. The analog signal produced by amplifier is usually converted into a digital signal and passed to a microprocessor. So, the data is processed, converted into a concentration units and output to display device or data is stored. Okay. So, this is a digital signal and the digital display includes digital signal production and passed to the microprocessor. From the microprocessor, the data produced is converted into concentration units in the sense the concentration of target analyte, and output is displayed in your display device or it is stored for further data analysis. So, we told there are two components, one is biological recognition component, another one is a physical transducer. So, what are all the kinds of various kinds of biological recognition element? So, one is enzyme based, another one is whole cell based, another one is affinity biomolecule based. The enzyme based or what are all the example? Catalytic transformation of specific substrates, right. So, in the presence of catalyst, how the specific substrate, which is nothing but a analyte, gets transformed, right. So, for example, phenol detection using tyrosinase, right. So, this tyrosinase is nothing but a we call it as a polyphenol oxidase, right. So, this is the enzyme which is nothing but a tyrosinase. So, what happens is you have a catechol, first phenol is converted into catechol. So, in presence of tyrosinase, so this catechol is converted into orthoquinone. Okay. So, this particular reaction is measured by the enzyme based biological recognition element. So, catechol is nothing but OH, okay. So, which converted into tyrosinase in the presence of tyrosinase. So, which is nothing but giving the oxygen, right. So, which is the oxidase, right. So, then which gives 2 so which we call it as a orthoquinone. So, what comes as 2H2O, okay. So, this is what the reaction is. So, if the phenol component is there in presence of tyrosinase, so as I told here, so this is the reaction pad. So, tyrosinase is kept in the reaction pad. So, substrate which is nothing but a phenol which comes and gets converted into orthoquinone. So, I will be able to measure this particular product. So, how much this product is there, that is proportional to the how much phenol is there in the particular food material, right. So, that is the way the phenol detection using tyrosinase is done. So, this is nothing but a enzyme based recognition. So, enzyme based recognition is this enzyme is kept in the electrode. For example, if you want to use a electrochemical transducer, right. So, for potentiometric electrode, in one electrode this particular enzyme is kept. So, when the electrode sees the target analyte which is nothing but a phenol. So, phenol comes and attaches in the tyrosinase. So, then it converts into orthoquinone. So, this conversion is detected as any one of the signal. For example, here it is detected as a electrochemical signal, then that is further fed into transducer, then electrical signal, then further processing is done. And another example is specific inhibition of enzyme activity by the target analyte. It is not only the target analyte comes and binds with the enzyme and produces the product, sometimes it is a specific inhibition also. For example, acetylcholine esterase inhibition by organophosphates, right. Acetylcholine esterase inhibition by organophosphates. So, these organophosphates are present toxins, right, which is present in the insecticides, okay. So, this organophosphates, right. So, this acetylcholine esterase is the enzyme, right. So, which converts the or which degrade the acetylcholine, right. So, the acetylcholine esterase degrades the or degrade it takes part the degradation reaction of acetylcholine. So, what happens is the organophosphates present in the insecticides goes and inhibit the activity of acetylcholine esterase. So, that is the reason the degradation of acetylcholine stops, right. So, the biological recognition element can be of specific inhibition of enzyme activity as well. So, the next one is effect of enzyme activity by the target analyte, which acts as a modulator of cofactor enzyme, right. So, here we talked about the phenol or catechol is converted into orthoquinone in the presence of tyrosinase, right. This tyrosinase can be activated by using cetine ions. It is not tyronase here exactly. So, I am telling this kind of enzymes can be activated by further modulator of cofactor enzyme. So, these mostly they are ions. So, your enzyme based recognition can be done in three ways. One is catalytic transformation of specific substrate, and another one is specific inhibition of enzyme activity by a target analyte, and another way it can be done is effect of enzyme activity by the target analyte, which acts as a modulator for coenzyme activity. So, how much target analyte is there, I will be able to measure by increase in activity of the particular enzyme, because the target analyte increases the activity of the particular enzyme. If the activity of the enzyme is increased, obviously the substrate to product conversion is going to be increased. The second recognition component is whole cell based. So, the general inhibition of cellular respiration, right, that is disturbed. So, in that way we will be able to measure the target analyte. And another one is analyte acting as a inducer or specific catalytic protein, right. If you are having a whole cell attached into the for example, here if you are using the potentiometric electrochemical transducer, this whole cell is attached into the electrode. So, the analyte which is going to be measured acting as a inducer of specific catalytic protein. So, based on the measurement of specific catalytic protein, I can measure the analyte. Then the third one is affinity biomolecule base. So, this is antibody antigen, ligand receptor, or sometimes nucleic acids, right. So, the hybridization of nucleic acid. So, based on these three varieties of biological recognition component, so biosensors were made. And physical transducer wise, we already discussed electrochemical, potentiometric, amperometric, calorimetric. Amperometric is based on redox or oxidation reaction, potentiometric is based on potential difference, calorimetric is based on the temperature difference. And optical optoelectronic sensors, light based potentiometric sensors, surface plasma resonance. So, this is based on the refractive index, and UV visible absorbance. So, you are all very well know based on the absorbance of the UV light. And luminescence and fluorescence we have already told. The total internal reflection, so TIRF, based on that as well the optical biosensors are made. The next one is piezoelectric. So, it is based on the quartz crystals, right, which converts the signal, quartz crystal microbalance. The next one is surface acoustic wave sensor. So, that is also comes under the category of piezoelectric transducer. The last category is thermal sensors, right. So, this is isothermal titration calorimetry. So, it comes also under the category of calorimetric electrochemical transducer. The next one is heat sensitive change in the polymer film color. So, this also can be used as a sensing element in the physical transducer. So, the broad category is either electrochemical signal is given to the transducer, or optical signal is given to the transducer, or piezoelectric signal is given to the transducer, or thermal signal is given to the transducer. So, this transducer further convert these signals into electrical signals. So, electrical signals are given to the amplifier, that is further amplified and given to the signal processing unit. From the signal processing unit, it goes to the digital processing unit. So, where it is sent to the microprocessor. So, from there we will get the digital display. So, now next one is the electrochemical biosensor. So, the electrochemical sensor where electrochemical signals are generated during biochemical reactions and monitored using suitable potentiometric, amperometric or conductometric system of analysis. So, all comes under, as we discussed here. So, all comes under the category of electrochemical transducer, potentiometric, amperometric as well as calorimetric. So, the electrochemical transducer is a one where electrochemical signals are generated during biochemical reactions and are monitored. So, these electrochemical signals are monitored using suitable potentiometric, amperometric or conductometric systems of analysis. So, it is considered as a chemically modified electrode since electronic conducting, semiconducting or ionic conducting material is coated with the biochemical film. So, that is what I told. For example, in terms of whole cell, right. So, the whole cell is kept in the electrode, right. So, that is what it is told. It is considered as a chemically modified electrode. It is not a normal electrode, it is a chemically modified electrode since electronic conducting, semiconducting or ionic conducting material is coated with the biochemical film. Many enzyme reactions such as those of urease and many biological membrane receptors may be monitored by a ion conductometric or impedometric devices using a interdigitated microelectrodes, as the sensitivity of the measurement is hindered by a parallel conductance of the sample solution. So, you are aware of that, right. So, in any biochemical reactions I take as a reference, right. So, here also as the sensitivity of the measurement is hindered by the parallel conductance of the sample solution, usually a differential measurement is preferred, but when a sensor with the enzyme and an identical one without enzyme, right. So, sensor with enzyme is, it is coated with the biochemical film, the one without enzyme is without any biochemical film. So, the comparative measurement is taken, it is not comparative measurement in the sense differential measurement. Analytes like urea, charged species and oligonucleotides are detected using this principle. So, mostly biochemical reactions we need to have a reference as well as the sample with the analyte solution. So, calorimetric biosensors measure the change in temperature of the solution containing the analyte following enzyme action and interpret it in terms of the analyte concentration in the solution. When the enzymatic reaction happens, most of the enzymatic reactions are exothermic. So, the heat is liberated. So, the system or calorimetric biosensor measure the change in temperature of the solution which contains the analyte following the enzyme reaction. Once the enzyme catalyte reaction has happened, so there will be a exothermic reaction. So, this exothermic reaction increases the heat, right. So, the heat generated by the reaction is used to determine the analyte. So, that temperature changes measured and interpreted in terms of analyte concentration. So, this much analyte is present in the solution. So, this much temperature increment will be there. So, calorimetric biosensor are extensively used for the detection of pesticides and other enzymatic reactions. The next one is potentiometric. So, potentiometric we have an indicator electrode and a reference electrode. A transducer may be an ion selective electrode which is an electrochemical sensor based on the thin films or selective membranes as the recognition elements. So, usually the potentiometric biosensor is something. So, we have a reference electrode, so which is of AG or AgCl. So, this is nothing but a reference electrode. Then this is filled with the internal aqueous solution. Okay, so that is filled, and here you have a salt bridge or membrane. So, this is nothing but a membrane. Okay. So, this is nothing but porous membrane. Okay. Yes, transducer may be an ion selective electrode, which is an electrochemical sensor based on thin films or selective membranes as a recognition element. So, this is nothing but a recognition element. So, which has the membrane in it, right, attached to it. So, the reference electrode is nothing but a Ag or AgCl. The most common potentiometric devices are pH electrodes, several other ion in the sense fluorine, iodine, cyanide, potassium, sodium and calcium NH4, these are all ions, right, or gas CO2 or NH3 selective electrodes are available. The most common potentiometric devices are pH electrodes. So, several other or gas or selective electrodes are also available. The potential difference between these indicator and the reference electrodes are proportional to the logarithmic of ion activity or gas concentration as described by the Nernst-Donen equation, right. So, the Nernst equation is something like E cell, which is equal to E naught cell minus RT ZF log of QR, right. So, this is nothing but the cell potential, right, and cell potential at standard conditions. R is gas constant, temperature, Z is nothing but number of electron transfer, F is Faraday's constant. So, QR is nothing but a quotient, nothing but reaction quotient. So, this Nernst-Donen equation uses this in equilibrium condition. So, Donen contributed in the equilibrium concentration. So, this particular thing is not necessary here. Here it is a mechanism is told, right. The potential difference between these indicator and, so this is the indicator and this is the reference electrode. The potential differences between them are proportional to the logarithmic of ionic activity. So, that is what I just told. So, the proportionality constant is here nothing but a standard E cell potential, right. So, or gas concentration, if you are using the ionic electrode, it is a ionic activity. If you are using the gas, then it is a gas concentration as described by the Nernst-Donen equation. So, this is a Nernst equation. This modified form is Nernst-Donen equation. So, I just wanted to tell you that it is a proportional to the logarithm of ionic activity. So, this we call it as a reaction quotient. This Q is nothing but a reaction quotient. Okay, so, then is amperometric biosensor. So, this is a first commercial biosensor designed by Leyland and Clark. So, this was done to monitor the glucose. The amperometry is based on the measurement of current resulting from electrochemical oxidation or reduction of electroactive species. It is usually performed by maintaining a constant potential at platinum or Au or carbon based working electrode or an array of electrodes with respect to the reference electrode, which may be also function as a auxiliary electrode if currents are low. So, you have a working electrode and one is reference electrode, right. So, if the current are low, for example 10 to 9 or 10 to 6 ampere, then auxiliary electrode is also used. So, this is nothing but your working electrode and one more is reference electrode. The resulting current is directly interrelated to the bulk concentration of the electroactive species or its production or consumption rate within the adjacent biocatalytic layer. So, this working electrode senses this bulk concentration of the electroactive species. So, then the resulting current between two electrodes is given as a amperometric signal. So, that is further converted into electrical signal using transducer. A biocatalytic reaction rates are mostly first order dependent on the bulk analyte concentration. This we have seen, right, all enzymatic reactions also of first order reaction. So, such steady currents are usually proportional to the bulk analyte concentration, right. So, these are first order based on the bulk analyte concentration. So, whatever the steady current produced are usually proportional to the bulk analyte concentration. So, amperometric biosensors are used in measuring sugars, alcohols, phenols, then oligonucleotides and oxygen. these are all can be measured using amperometric biosensor. So, it is a single enzyme based also there, multi enzyme based also there. So, a single enzyme based the example is acetyl choline esterase. So, what we have discussed, and there are butyryl choline esterase also there. So, if you remember acetyl choline esterase we have discussed here in terms of the organophosphates, right. So, as a single enzyme as a biological component, and thiocholine production is monitored amperometrically or acid production is monitored potentiometrically. So, the product based on that you can have any measurement, right. So, the thiocholine production also you can monitor using amperometrically, or acid production is monitored using potentiometrically. So, this is like using a single enzyme based biosensor, either how much substrate is consumed, that also can be measured, or how much product is produced, that also can be measured. Multi-enzyme based biosensor system is also available, which uses the choline esterase in conjunction with the choline oxidase, both the enzymes are there, right, and measure the hydrogen peroxide production or oxygen consumption. So, if oxygen consumption is there, there may be a hydrogen peroxide, right, that is what we call it as a oxidation reaction. So, either it can be measured as a production of hydrogen peroxide or consumption of oxygen. So, this uses the multi enzyme. So, one is choline esterase, another one is choline oxidase, both. Then optical biosensor, which are called optodes instead of electrodes. So, they are resemblance with the electrodes. These include determining changes in the light absorption between the reactants and the products of the reaction, measuring the light output by a luminescent process. So, luminescent process already I told you. The optical biosensor integrate optical technique with the biological element to identify chemical or biological species. Many optical sensors were developed based on the surface plasmon resonance spectroscopy, UV spectroscopy, we are very much known to that. Then evanescent waves, right. It monitors, optical biosensor is used to monitor pesticides, vitamins, carcinogens, toxins based on chemiluminescence or fluorescence technique. So, optical biosensors are similar to electrode, but here it uses the light absorption between the reactants and products to measure the analyte concentration using a luminescent process. So, it can be measured using spectroscopic or evanescent waves or surface plasmon resonance. So, it monitors pesticides, vitamins, carcinogens and toxins based on chemiluminescence or fluorescence technique. Then there comes a microbial biosensor. So, as I told earlier in previous slides, in an analytical device which combines the microorganisms with the transducer to facilitate the rapid, accurate, sensitive detection of target. Right. So, as I told, in the reaction pad I kept the enzyme, the substrate comes and binds and goes a product. So, instead of the particular enzyme, there can be a microbial cells which also can be attached. So, that is what it says, combines the microorganisms with the transducer to facilitate rapid, accurate and sensitive detection of target. So, conventional microbial biosensor used the respiratory and metabolic functions of the microorganisms to detect a substance that is either a substrate or inhibitor of the process. For example, as I told, if substrate gets converted using particular enzyme. So, that particular enzyme is produced by these microorganisms which is attached to the transducer. So, it uses, how it detects, it uses either respiratory or metabolic functions. Metabolic functions in the sense microorganisms produces the enzyme, that is the enzyme which converts the substrate into product, right. So, this can be measured in this way or just by respiration activities of the microorganisms, and whether that is inhibited or activated by the analyte concentration, that way also the analyte concentration can be measured. Microbial biosensors are more advantageous than enzymes biosensor. Why? Because the construction of enzyme sensors are complex and costly, because we need to be very much specific about that particular enzyme. Some of the main types of microbial biosensor, it can operate in an amperometric way, potentiometric way or conductometric way. So, that means it can be analyzed based on oxidation and redox reaction, or it may be analyzed based on the potential differences, or it may be analyzed based on the conductivity of the material differences. Amperometric microbial biosensor operates at a fixed potential with respect to a reference electrode and involves the detection of the current generated by the oxidation or reduction of the species at the surface of the electrode. This I told. Amperometric microbial sensor are extensively used for detection of biological oxygen demand BOD in industrial wastewater, and also used for detection of ethanol, total sugars, organophosphates, cyanide, phenols and phenolic compounds. Generally, potentiometric microbial sensor consists of an ion selective electrode that we have seen somewhere here, right. So, this one. So, or a gas sensing electrode coated with an immobilized microbe layer. So, it is a microbial biosensor. So, it is coated with the immobilized microbial layer. Due to microbial metabolism, the uptake or release of analyte generates a change in potential resulting from an ion accumulation or depletion. The potentiometric transducers measure the potential difference between the working electrode and reference electrode. The signal is correlated with the concentration of analyte. So, that is common everywhere. The signals are expressed in the logarithmic relationship between the potential generated and analyte concentration. So, wide detection range is possible. The potentiometric microbial sensors are used for the detection of organophosphates, penicillin, tryptophan, urea, trichloroethylene, ethanol, sucrose. Whole cell biosensor immobilization of Pseudomonas alkaligenase MTTC 5264 having the capability to degrade caffeine were used for the development of caffeine biosensor. In the caffeine biosensor, Pseudomonas alkaligenase, this is used as a microbial film, right. So, what we are doing here, we are immobilizing the microbial layer in the electrodes. So, the immobilization of Pseudomonas alkaligenase. So, that this is done in the electrode to check the degradation of caffeine in the development of caffeine biosensor. The biosensor system was able to detect caffeine in the solution over the concentration range of 0.1 to 1 milligrams per ml. So, this is the example for potentiometric microbial biosensors. So, immunosensors, we will see this as an example. The immunosensors are affinity ligand based biosensors. So, this I told you have to have a receptor and ligand. Biosensor devices in which the immunochemical reaction is coupled to a transducer. They are solid state devices. So, these immunosensors are solid state devices. The fundamental basis of all immunosensors is the specificity of the molecular recognition of antigens by a antibody to form a stable complex which is similar to the immunoassay methodology what we use in biochemical detection. So, the immunosensors can be classified based on the detection principle, again the electrochemical, optical or microgravimetric. So, based on that we have seen already. In contrast to immunoassay, modern transducer technology enables the label free detection and quantification of the immune complex. So, how we are going to see how it can be measured. Immunobiosensor based on chemiluminescence for detection of vitamin B12. So, that we are going to see. So, this is as an example. So, how the modern transducer technology enables the label free detection, quantification of the immune complex. So, the example what we are going to see is vitamin B12 detection. So, here is your dipstick spotted with the antibody. right. So, this we call it as a dipstick. So, this particular thing is a dipstick. So, here in the end we have a antibody. So, this end looks like this. So, you have a antibody. So, Y shaped protein, antibody coated on a NC membrane. So, nitrocellulose membrane. So, this upon washing, after washing that, this is dipped into vitamin B12 solution, right. So, this antibody binding with the vitamin B12. So, this all red mark, right. So, this red mark is nothing but a vitamin B12. So, vitamin B12 is binding with the antibodies, then upon further washing, then this is dipped into a ALP, right, alkaline phosphate conjugate solution. So, this is dipped into vitamin B12 ALP conjugate solution. So, then binding of vitamin B12 ALP conjugate with antibody. So, this star is blue star is nothing but vitamin B12 ALP conjugate. So, then further it is dipped into cuvette containing substrate CDP star. So, this substrate is nothing but a luminance property material. So, this substrate CDP star. So, once it is dipped into this particular cuvette, so it emission of Cl due to reaction of ALP conjugate with the CDP star. So, ALP conjugate with the CDP star. So, it emits the light, right, chemiluminescence. So, Cl is nothing but chemiluminescence, chemiluminescence emission. So, this particular star is nothing but a chemiluminescence emission. So, this is Y is antibody. So, once it emits the chemiluminescence, that is detected by the luminometer, then it further go to data analyzer. So, what we are seeing here, so first the dipstick with antibody is kept in the vitamin B12 solution. So, the vitamin B12 is getting attached with the antibody. So, then further it is dipped into vitamin B12 ALP conjugate solution. So, then ALP conjugate sits in the vitamin B12 and antibody combination. Then further it is dipped into the substrate CDP star. So, then it emits the chemiluminescence due to the reaction between the ALP and CDP star. So, how much chemiluminescence is there, that much ALP is there, how much ALP is there, that many B12 is there. So, that is the way the concentration of B12 is measured using luminescence technology. So, this is the CDP star. So, upon ALP, so it forms the unstable intermediate. So, this unstable intermediate further gives one particular component, another component which emits the light. So, this is the way the vitamin B12 concentration is measured using chemiluminescence emission. So, here I stop. Okay, so these are references and additional resources for this particular lecture. So, you must be wondering, so each lecture got some extra resources as references. Actually, these 10 lectures are about recent topics in food processing. So, I try to include recent research developments also in this lecture. So, that is why mentioning where I have taken the recent research developments in particular lecture. So, these three and most of the points were taken from this particular review article. Okay, so, I would also request you to check these extra references to get to know more about that particular topics. Thank you.